Today we will learn about vaccines. An individual exposed to an antigen is the foreign substance which may be virus, bacteria, to induce formation of antibody. Antibodies are that produced against the antigen, are the proteins that protects the body. A type of immunity known as artificial active immunity, material used to induce artificial active immunity, the antigen or mixture of antigen known as vaccine or immunogen, and process of generating such immune responses immunization, and commonly known as vaccination. It derived from vaccinus, meaning from cows. It is a non-toxic or non-virulent preparation of antigenic material that can induce long-term humoral as well as cell-mediated immune response against pathogen. Now here major difference between humoral and cell-mediated is humoral protects from outside or extracellular, and cell-mediated protects from intracellular toxins. Now here types of antigen used in vaccine. Anything from whole organism to small subcellular fragment can be used as antigen in vaccines. Most current vaccine described as whole organism which include live but attenuated means used weak form of the germ, or killed organism as subunit vaccine, toxoid, capsular, polysaccharide and recombinant surface antigen. Now live but attenuated vaccine, prepared by attenuating pathogenic organism by growing them in unfavorable condition, which result in gene mutation due to which organism loses pathogenicity but retains their capacity for transient growth. It has advantages as well as disadvantages. Due to their capacity for transient growth, these vaccines have prolonged immunogenicity and eliminates the need for repeated boosters. But a major disadvantage is that they associated risk back to virulent form. But these days genetic engineering also used to cause. Now inactivated or killed vaccines produced by inactivation of pathogenic organism by heat or chemical treatment. Organisms are unable to multiply in the host. Inactivation is carried out so that structure of epitopes are the structure found in the to which antibodies are bound. On surface antigen remain maintained. Hence, heat treatment is generally not satisfactory since it can cause protein degradation and loss of some epitopes. Thus, chemical inactivation using formaldehyde or other alkylating agent is common. Inactivated are effective, but they are less immunogenic, so often require several boosters, and normally do not adequately stimulate cell-mediated immunity or secretory IgA production. In contrast, attenuated vaccines are given in single dose and stimulate both humoral and cell-mediated immunity. Now, subunit vaccines. These are as toxoid, capsular, recombinase, microbial technology. Many exotoxin modified so that the antigenicity but no longer toxins is modified exotoxin is called as toxoid. Are not as efficient as original exotoxin but can give safely as in high dose. DNA vaccines are genetic vaccine. Use genetic material itself to immunize individual. Vaccine fragment genome encoding antigenic protein are injected directly into host so they can integrate into chromosomal DNA or exist as episomes. Episomes are self-replicating, for example, like plasmid. Expression of gene within host generate foreign protein to which host immune system responds. DNA vaccine is really an immune response made against the protein encoded by vaccine DNA. The DNA itself is not immunogenic. Now, recombinant vector vaccine that encode antigen isolated from pathogen can insert it into non-viral virus or bacteria. Such serve as vector replicating within host and expressing product of pathogen encoded antigenic protein. The antigen eliminates humoral immunity as well as cellular immunity.